Assalamu alaikum, everyone. I hope you all are doing well and you all are fine. So today's lecture is going to be about hematopoiesis and cell types. So yeah, we're going to first start with what is the definition of hematopoiesis? So it's a process by which the blood cells are formed in the body, and they are a complex process that involves differentiation of hematopoietic stem cells into various types of blood cells like lymphocyte show, monocyte show, like all the white blood cells, red blood cells, platelets. And this process to the asal ki bone marrow ki occur kigi, but it can also in adults, but in children and during fetal development it can occur in the liver and also occur in the spleen as well. So that's just the basic definition of it. And stem cells, so usually stem cells, these are a type of unspecialized cells that can differentiate into specialized cells, and they have the ability to self-renew themselves through cell divisions. And these cells can be found in various parts of the body. So the bone marrow, so such as like the hematopoietic stem cells, they are also found in the adipose tissue, and they're also found in the embryonic tissues. And as I mentioned, example, in bone marrow, it's mainly the hematopoietic stem cells, we're going to talk about, and they're responsible for production of all the different types of blood cells. And these stem cells, they actually play a very important role in clinically as well in medicine and science. Like they do ستم استماليجي that stem cells جدي like ستم استماليجي bone marrow transplantation تم استمالكيجي is da غير treatment غوند like bone marrow transplantation like leukemia sta lymphoma sta غسي blood cancers that جدي blood oncology كده دي important role لري. And also in other aspects of medicine and drugs as well أغيكم da important role لري. So the process of this is that it usually the blood cells they begin their lives in the bone marrow from a single type of cell, which is called the pluripotent cell. So it starts. So this is the main cell where it starts from, and from this all the cells that are circulating in the blood are derived. And as you can see here in this figure, you can see the successive divisions of this pluripotential cell that forms the different circulating blood cells. And as these cells reproduce, a small portion of them remain exactly like the original pluripotent cells, and they're retained in the bone marrow to maintain supply of it. And although their number usually diminishes with age, but most of the reproduced cells, however, differentiate to form other types of cells. So that tool hematopoietic stem cell that pluripotent stem cell ورتوي or that أصل كبير زان differentiate كي into erythrocytes, into platelets, into neutrophils, monocytes, eosinophils, basophils, plasma, B lymphocytes, T lymphocyte, that tool daga cells da sawarki, the daga cells data psaki, juri gi pedaki, so they all grow from this. And what happens is that during hematopoiesis, the hematopoietic stem cells are mainly multipotent stem cells that I work. So are lymphoid stem cells that how do you say it? They form into the lymphoid stem cells, and they also form into the myeloid stem cells. And the myeloid progenitor cells, they didn't give rise to red blood cells, platelets, as you can see here, and they also give rise to the white blood cells like neutrophils show, like monocytes show. And the lymphoid progenitor cells, it will give rise to B lymphocytes and also T lymphocytes. And the different committed stem cells will also produce like colonies of specific types of blood cells. So example, a committed stem cell that produces erythrocyte, this is a colony forming unit, erythrocyte. And likewise, colony forming unit of granulocytes and monocytes also are present here. So And as these progenitor cells, they become more specialized. What happens is that they lose their ability to differentiate into other types of blood cells. So asal da pluripotent cell da lymphoid stem cell dina peda kigi da be myeloid stem cell ki ham badligi, and the myeloid stem cell da further on da colony fodita da committed stem cell ki badligi da basically it differentiate into colony forming unit erythrocyte that myeloid the colony forming unit erythrocyte the badligi, and da through this process, so through further differentiation, further specialization of the cells, the further down to the erythrocyte the badligi, and then here it also forms a colony forming unit megakaryocyte over time further down the specialized kigi or differentiate kigi platelets well colony forming unit, the granulocytes and myocytes when they go through all these stages of differentiation and all these stages of specialization, and eventually with neutrophils form monocytes, eosinophils, basophils, and then the lymphoid stem cell da badlig da pre B cells and pre T cells and then further on the lymphoblast and T lymphoblast and eventually the lymphocytes and T lymphocytes in a jurigi. So this is the general rule for this. So usually what happens is that these committed stem cells, they are actually characterized by the expression of specific surface markers, which can be used to identify them. For example, the myeloid progenitor cells, we express CD34, CD38, and CD117, whereas the lymphoid progenitor cells, we express CD34 and CD10, and also CD19. And what it does is that these CD markers, which is also called cluster of differentiation, and they are specific. These are cell surface proteins that are specific for these cells. And the expression of these markers can actually help to distinguish between these different types of progenitors. And as I mentioned, can also be useful in diagnostics and research settings as well. And also in terms of regulation, the process of commitment is tightly regulated by a variety of signaling molecule. So the whole process to the control by signal molecule like a cytokine the growth factors with the process, the hematopoiesis process we control. So what they do is that these molecules, they will bind to specific receptors on the surface of these progenitor cells, and they will then trigger a series of intracellular signaling events, like example, they will trigger activation of transcription factors, and then it will eventually then promote cell differentiation and maturation as well. And what happens is that the regulation of the commitment is actually critical for maintaining a balance between the different types of blood cells, and also preventing the development of blood disorders. So formation of these growth inducers, so these cytokines and these growth factors, and it's actually controlled by factors which is outside the bone marrow. So for instance, example, in the case of erythrocytes, if there's exposure of blood to low level of oxygen for a long time, what it does is that it causes growth induction and differentiation, and then production of greatly increased number of erythrocytes. And this is going to be discussed like in down the slides in more detail. And also example, in the case of some of the white blood cells, so example, infectious diseases can cause further increased growth and differentiation, and eventually formation of these specific types of white blood cells that are needed to combat the infections. So that's what this is. Okay. So this is just a description of what I just explained about the signaling molecules and cytokines involvement. So there are different types of leukocyte. Basically means as another word for white blood cells, and these are diverse group of cells that play a critical role in the immune system. and there are different types of leukocytes, so each of them have unique characteristics and function. So that we have for the leukocyte the white blood cells or the agranulocytes or the granulocytes like neutrophils eosinophils basophils granulocytes because the granules the dots so that whole granules and granules are we like like vesicles that contains enzymes. The granules like vesicles enzymes contain and substances contain immune response yeah, so that's what they so basically we have like polymorphonuclear neutrophils. We have also the eosinophils, the basophils, and then there's a monocytes I mentioned, lymphocytes that are agranulocytes. These are known as agranulocytes, and also occasionally plasma cells too. And they're also, as I said, they're also called like polymorphonuclear cells. They all have like granular appearance, and this is why we can call them granulocytes. And they're called like polymorphonuclear cells because they do they have many nuclei. They have many nuclei. They don't have only one nucleus. They have nuclei, so multiple nuclei. They have like lobules of nuclei. And they protect the body against invading organisms, and mainly they're ingesting them by the process of phagocytosis, especially the neutrophils. So types of leukocytes. So with neutrophils, these are the most abundant type of leukocytes, and they make up about like 60-70% of total white blood cell count. They're the first responders to infections, and they're attracted to sites of inflammation. Neutrophils, these are also characterized by their multi-lobe nuclei and their ability to engulf and destroy invading microorganisms through phagocytosis. So neutrophils they do with the total white blood cell count the arish beta to a percentage the neutrophils دي per حساب أول cell دي involve the immune response involve the and through the process of phagocytosis. So yeah. And then there's lymphocytes, or the lymphocytes are like 20-25% of a total white blood cell count. They account for like 20-25% of total white blood cell count, and they refer to B cells, T cells, and natural killer cells. So B cells producing antibodies, which are proteins that recognize and neutralize specific pathogens. And there's T cells, on the other hand, which directly infect cells and coordinate in the immune response. So lymphocytes lymphocytes category B cells razi, T cells razi, natural killer cells razi. So B cells, as we know, that are we like antibodies produced are involved in like humoral immune response. And then there's also T cells that are involved usually in the cell mediated. And then there's also monocytes, and the monocytes, they make up like 3-8% of the total white blood cell count, and they're usually characterized largely by kidney-shaped nuclei, and they're the largest type of leukocytes, and that are able to engulf and destroy invading microorganisms through phagocytosis. And they can also differentiate into macrophages. So when they so inside the tissues, they can differentiate into macrophages, which are also specialized type of cells, And then there's eosinophils. They make up like 1-3% of total white blood cell count, and they're usually bilobed nuclei, as you can see here. So eosinophils, they have 2 lobes of nuclei here and here, just attached by a chromatin thread. And they're involved again like parasitic infections, and play a role also in allergic reactions too. Then there's basophils. They make up less than 1% of total white blood cell count, and they have a large granular cytoplasm, and they usually release histamines, which involve inflammatory response and play a role in allergic reactions too. So this is basophils, and you can see that the nucleus, the cytoplasm is large, and it's very granular as well. So here is like the monocytes, and as you mentioned, they're found in the blood, differentiate into macrophages. and so macrophages چې دي کله چې دا monocytes په tissue کې then هغوی چې دي څه کېږي هغوی macrophages ته بدلي کله چې circulating in the blood they're known as the monocytes and they also function as antigen presenting cell via MHC2 molecules and cell surface proteins. So what MHC mean is that MHC these are known as the major histocompatibility complex, and this is a set of genes that code for cell surface proteins that are important in the immune system. So MHC these are divided into MHC1 and MHC2. So MHC these are divided into MHC1 molecules and MHC cell surface proteins MHC2 دي نوم چې د موږ major histocompatibility molecule ورته وایو class 1 چې دا هغه they're found on the cell surface of all the nucleated cells in the body, and they present antigens to cytotoxic T cells, whereas MHC class 2 molecules on the other hand they're found mainly on the specialized antigen presenting cells like dendritic cell shoe, macrophages shoe, B cell shoe, and they present antigens to helper T cells. So these are very important. And then and then there's the macrophages culture macrophages specific tissues ki example liver ki هغوی خپل specific names لري so liver ki دې ته موږ Kupffer cells وایو and په connective tissue ki دې ته موږ histiocytes وایو په bone ki ورته osteoclasts ویل کیږي and په brain ki ورته microglial cells ویل کیږي. And then we have got the dendritic cells. These are antigen presenting cells. The innate immune system and the adaptive immune system. Your both are the connect. And the dendritic cells dwar MHC class 1 basically present MHC class 1 NFC receptors. And then eosinophils, as we discussed, that they're very important for parasitic infections and also for like asthma and all like chronic adrenal insufficiency, all these ones, allergic processes too. And basophils, again, allergic reactions, and it's that the basophils the total white blood cells the basophils and the eosinophils come compared to the other ones. And mast cells like we discussed in asthma that it's important and how important it is in asthma and how important in type 1 hypersensitivity reactions. So mast cell type 1 hypersensitivity reactions can involve kigi or the amaga precursor cell na originate kigi community basophils originate kigi but during hematopoiesis kichida. And so lymphocytes we mentioned again matlab yani refers to B cells, T cells, natural killer cells, and mediate adaptive immunity as well. So natural killer cells they are larger than B and T cells, and they contain lytic granules. So first of all you can see the CDs that's here, this one and these ones and these ones. I'm literally covering them all. These ones. So data today kana munga cluster of differentiation wayu, and this is basically a classification system that is used to identify and characterize cell surface molecules on various cell types, and each CD molecule is associated with a specific function and a role in the immune system. So that classification system that associates surface molecule that is specific different cells that protein that they're specific for various types of cells, and the specific function or the specific role immune system so they're very important, and they also help in identifying the type of cell. So that cell example identify for example natural killer cell through CD56 and I through these and B cells through CD20 yeah CD19 through DAGA identify KIGI usually, and same with the T cells as well, so these are important too. And coming back to natural killer cells, these are important in inducing apoptosis. So what they do is that they bind to surface receptors of the infected cell and the surface proteins. And what they do is that they then release their granules, these lytic granules, and they then induce apoptosis in the infected cells or in the abnormal cell, basically. So the natural killer chidi Canada base up like infected cells kida say sakigi bind kigi da sulfur receptors the bind kigi achakala bind shi khpal lytic granules be release ki, and the lytic granules chidi aga apoptosis induce ki matlab cell death induce ki aga infected and virally infected cells ki or abnormal cell ki. And mentioned again B cells chidi the humoral immune response initiate ki. और B cells चीदी असल के मुंग चंगा hematopoiesis के slide के ओकतल ची B cell we originate कीगी bone marrow के originate कीगी लेकिन and mature कीगी हागटीगी यानी bone marrow की दीदो produce कीगी अमलता bone marrow के गटीगी हम लेकिन कल after maturity B cells चीदी दीदो बे migrate के अखबल secondary lymphoid tissues ta like lymph nodes ta migrate कई spleen ta migrate कई encapsulated lymphoid tissues ta migrate कई. And what happens kala chibe antigen ya antigen chikala da B cell encounter ki da B cell chidi kan zan plasma cells ta badli antibodies produce kai and zan ham memory cells ta ham badla shi, and T cells chidi the cellular immune response ke role lari. So they originally again the ham lama lymphoid the progenitor chida lymphoid multiple lymphoid sadam lymphoid stem cells that are razi, and then the hamamalta bone marrow ki produce kigi, but the maturity jada gatwale da thymus ke occur kigi. So they mature, this maturity it occurs in the thymus, and there's a different types of T cells. So we have cytotoxic T cells which express CD8. The CD8 jada they specific the cytotoxic T cells da, and then we also another type is helper T cells which express CD4 or the CD4 jada the specific the helper T cell, and then there's also regulatory T cells CD28 bande the cell that is specific so as I mentioned that CD the cluster of differentiation way or their classification system so cell surface molecules the specific cells ke. And again, plasma cells we produce antibodies, and they're found mainly in the bone marrow, and they normally do not circulate in the peripheral blood. So if you know, it's basically involved in the pathology of multiple myeloma. So as we can see, the locations of this. So we mentioned that the spleen is the site for these are the secondary lymphoid tissues, and this pays patches as well. Let me just go. There's another. So yeah, and then lymphatic vessels. So what happens is so granulocytes and monocytes, as we mentioned, we bone marrow ki juri lymphocytes, plasma cells chidi we produce ki bone marrow ki, lekin kala mature ki ga lymphogenous tissues ki mature ki like lymph glands show, spleen show, thymus show. Thymus gland show, a tonsil show. So these are basically secondary lymphoid tissues that's elsewhere in the body, and also so the other white blood cells like monocytes and granulocytes, we have bone marrow ki juri jamal to store ki until they're needed in the circulatory system. And yes, so and then the composition of blood the blood zumunga, it's composed of 55% of plasma, 1% of these buffy cochimogorto, and it consists of white blood cells, platelets. So and then erythrocyte, which makes like around 45% of the blood, and then there is like yeah. And then there's this is the process of erythropoiesis, specifically how red blood cell is being formed. specifically. So as you mentioned, that hematopoietic stem cells should are we in first set differentiate key. So differentiate into pro-erythroblast difference key بدليجي ببرو erythroblast ديدو early erythroblast سكيجي transfer سكيجي بدليجي, and at this stage شدا, so first it starts with pro-erythroblast, then it's early erythroblast. This is how the red blood cells are being matured. So it starts with a pluripotent hematopoietic stem cells, then it starts with myeloid multipotent stem cells, multipotent myeloid stem cells, then it forms into colony forming erythrocytes, and then further on amaga that differentiate key into pro-erythroblast, then early erythroblast, and then at this point and then into late erythroblast, and at the point of late erythroblast فدغه point کې چې دا دغه point کې دلته hemoglobin ځان accumulate کوي, and then the late erythroblast develop into normoblast, which will then become reticulocyte, and at this stage, as you can see, the nucleus is being lost, and once the reticulocyte enters the circulation from the bone marrow, like after like about 24 to 48 hours, it will then mature into erythrocytes, and erythrocytes usually has a lifespan of 120 days, and before it's being removed from the circulation. So yeah, this is the process of that. And normally what happens is that about 90% of all the erythropoietin is being formed in the kidney. So erythropoietin, it's a hormone that stimulates the production of red blood cells. And what it does, that when a person, example, is placed in an atmosphere of low oxygen, what happens is that the kidneys, you can see in this diagram here, the kidneys, they will signal for the secretion of erythropoietin, and erythropoietin will then stimulate the production of pro-erythroblasts. So the hormone chida erythropoietin چې مونږ ورته وایي دا hormone دا چې کله release شي into the blood stream, because kidneys like low blood oxygen notice کوي, and factors that decrease oxygenation could be like low blood volume, anemias, low hemoglobin, poor blood flow, or even pulmonary diseases. هغه ټول چې د هغه kidneys په حساب sense کوي, and kidney چې د signal send کوي چې د هغې د وجې نه هغه erythropoietin secrete کړي. کله چې erythropoietin secrete شو, نو هغه erythropoietin دا hormone دا چې کله release شو, نو دا stimulate کوي production of red blood cell. So it stimulates this process. And what happens is that in addition to this, once the pro-erythroblasts are formed, the erythropoietin then causes these cells to pass more rapidly through the different erythroblastic stages than they normally do, so further speeding up the production of new blood cells. So what happens is that kala چې دا erythropoietin kala produce شو, دا process speed up کوي. دا process چې دا because of erythropoietin چې دا process that is speed up کوي. So they can because of low oxygen, low level of oxygen, to increase the production of new red blood cells. So the rapid production of cells then will continue for as long as the person remains in a low oxygen state, or until enough red blood cells have been produced to carry enough amount of oxygen to the tissues. And at this time, the rate of erythropoietin production then decreases to a level that will maintain the required number of red cells, but not at an excess. So in the absence of erythropoietin, few red blood cells are formed by the bone marrow. So as I mentioned, the erythropoietin, the rate of erythropoiesis increase کوي کله چې blood supply, کله چې oxygen level کمی, کله چې kidneys زمونږ low oxygen notice کی, and then when the level becomes normal, the process چې دا it also decreases. So this is دا your negative feedback so the process, the negative feedback mechanism so the process دا. And then we have the platelets. So platelets, these are also known as the thrombocytes, and we have مونقد the platelets بمورد كنور lectures هم لرو. So آغي كده platelets concept deal per detail كي explain كيجي. And platelets كده, these are thrombocytes, are small disc-shaped cell fragments that are involved in blood clotting, and these are formed through fragmentation of megakaryocytes, which are large polypoid cells found in the bone marrow. And megakaryocytes, what they do is they will produce proplatelets, which then extends into the bloodstream and will undergo fragmentation to produce small discoid platelets. And these platelets are actually, as mentioned, are essential for blood clotting and are activated in response to tissue injury or tissue damage. And the process of platelet formation is actually tightly regulated and involves the coordinated action of various cytokines and again growth factors. So again, the production of kind of stimulated when it's needed is by thrombopoietin hormone. So pasanga red blood cells ke erythropoietino, but they ke thrombopoietin that the platelet formation production and stimulate K, and it acts by stimulating the growth and maturation of megakaryocytes, and which produce the plates eventually. And again, this is also a negative feedback mechanism. So myeloid stem cells should be the megakaryoblast and through this megakaryocytes, and then the fragment kigi into platelets, which are then released into the bloodstream, and it's being stimulated by the hormone or regulated by the hormone thrombopoietin that's being released from the kidneys and the liver. So that's all for today's lecture. It was very brief,